For this lesson, I'm going to cover paper 5 on the planning skill, and I'm going to split the lesson into 2 parts. In part 1, for today's video, I'm going to show you the guide on how to plan for paper 5. In my second video, I will show you another sample which can actually score a higher mark and an alternative writing method. So I will guide you on the skills of paper 5 planning with reference to this past year question, which is May June 15 question paper 52, question 1. So for all planning questions, before they start, they will usually tell you the aim of the experiment. So for this experiment, students actually, they're trying to make a solution of enzyme extract. And this extract contains the enzyme amylase. So what is the purpose of this enzyme amylase? They are planning to find out the concentration of amylase in the extract. To identify the concentration of the unknown amylase in the extract, I will need to prepare a series of standard solution using the stock solution, where I can see that the concentration is 0.5 gram per dm cube. And then I will need to have starch, which is actually the substrate, which is prepared into a petri dish. So now I can tell that this is my independent variable, which is the concentration of amylase. So let's identify what is the standard variable. So in this experiment, we will see that the student is using petri dish plate to find out the concentration of amylase. We can see that they are using starch agar. So here you can think of like, most likely I need to standardize this. So SV represents standard variable. And since I'm using enzyme solution, most likely this one needs to be standardized in terms of the volume. And as the plates were incubated, most likely this one also I need to standardize the timing itself. And as I flood the plate with iodine solution, so some students might think that this is necessary to standardize the volume as well. But if this experiment is meant to judge color change, then the volume or the concentration of starch solution will be important, but not in this case, because by looking at the result, I would know that most likely the area stained brown, which indicates that starch has disappeared, will be my dependent variable. So I'll need to measure the diameter of the area stained brown. So from the diagram or from the question itself, I will most probably be able to identify a few variables that I know that I need to include into my planning experiment. Now I'm going to guide you on the rule of thumb in planning experiment. Number 1, usually we'll identify the independent variable. And the rule here is we will need to set up at least 5 sets. So identify from the question if the independent variable, if it's about a concentration. So if it's concentration, make sure you know how to elaborate it, where the 5 different concentration can be prepared using simple or serial dilution. If it's temperature, you might need to set it up with thermostatically controlled water bath. Or it could be pH, where you can prepare using buffer solution. Then number 2, you need to identify the standard variable in the question, and most likely is more than one variable. So if one of your factor is the independent variable, the other factors that is affecting the rate will be the standard variable. So your standard variable, for example, in an enzyme catalyzed reaction, it could be the volume of the solution, but you need to state specifically what solution. So it could be your enzyme, it could be your substrate, it could be your reagent, or it can be your temperature. So you must state how you control the temperature. It could be a pH. It can be your type or mass or the age of your specimen. And one of the main things that you can or need to standardize will be about the timing. So what are the timing that you can or need to standardize? Reaction time, incubation time, soaking time, or equilibration timing. So anything that could possibly affect your reaction needs to be standardized. Step 3, we can include some procedure that is exclusive for this particular experiment, such as if you have anything to do with gas, you make sure that it's airtight. Any experiment to do with plants that you have to cut the shoot and soak in water, you make sure that you cut the plant underwater to prevent air bubbles from entering. And if you have any experiment like for respirometer, we need to put in a soda lime or KOH that is meant to absorb CO2. And as for experiment to do with movement of indicator or dye, you can mention that your dye or indicator are placed at the beginning of the experiment or at the end of the tube. So this procedure will be very specific to the experiment. So there are few experiments that you have to memorize the procedure, such as chromatography, as like how you can place the spots and how you would use a fine pin to transfer the spots and how you place the paper into the solvent. And electrophoresis are also very specific experiment that requires specific procedure. Then number 4, we can actually talk about dependent variable. So after I've set up all the experiment, now it's time to record the result. So when you're recording the result, you make sure you state the endpoint. So your endpoint could be about your color. It could be about time. So you, for example, like Benedict test, you can say that record the time taken for the first color to appear. Or in terms of respirometer or sometimes photosyntometer, you can record the distance traveled within the fixed time. So you must always mention the endpoint and state specifically the timing or the distance or the color that you would want to actually identify. At the end of the experiment, do not forget to mention that repeat the experiment for the other independent variables. So if you have 5 different concentrations, you would mention that repeat the experiment for the concentration 2, 3, 4, and 5. And then for point number 5 to score, we will include reliability. How do you make sure that your experiment is reliable? So again here, you have to mention repeat the experiment, but this is for the whole procedure. For each concentration, you have to do it for another 2 times at least. So you will have a total of 3 results for each independent variable. So we will say, replicate the experiment for another 2 times for each independent variable and calculate the mean. For point number 6, we will relate to hazards and safety, where we would comment about the level of hazard, such as you can talk about if it's either a low, medium, or high risk. So despite its low risk for most of bio experiment, but we can still comment about the hazard where anything that could be an irritant or that can cause allergic reaction. So you can protect yourself by wearing gloves and safety goggles. And finally, if applicable, we can set up a control experiment. So this experiment, we would remove the independent variable or remove the factor that contributes to result. So this is meant to prove that whatever result that you obtain from the experiment, it's purely due to the independent variable that you're changing and not anything else. Or to show that the experiment result is really from the factor that you're trying to investigate and nothing else.
That's all for today's video. If you are interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with 1 or 2 past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.